Good afternoon, everyone. We did our project on gene expression, specifically the Gemini virus. And I'm going to let my group introduce ourselves. I'm Kelsey Gallant.
I'm Michael Kearney.
I'm Katrina Brown.
And I'm Nicholas Munson.
And we're short too. We also had Joseph and Quentin, but they couldn't make it today. So what is the Gemini virus? The Gemini virus is part of a family of viruses called the Gemini viridae, and it's basically a virus that is transmitted from white flies and crickets. And what it does, it changes the genetic encoding, so basically it negatively mutates the plant so it doesn't grow well, which destroys it.
The top picture is the healthy plant, and the one on the bottom right is one that's affected by the virus. Alright, saving crops around the world. That is our end goal with the research topic we were going with. See, we've used and studied gene expression. And gene expression is basically the way you saw the plant dying earlier. That's the genes expressing that because the virus came in and messed around with them. So we studied gene expression to see what the virus is actually changing in the genes of the plant so we can counterbalance whatever changes it's making and make future plants immune. Therefore, saving lots and lots of crops, including here in North Carolina with different plants, peppers, spices, and that stuff, and all over the world, making more food available to everyone.
Details. The plant that we use is called Arabidopsis thaliana, and we use it because we have a full genome already sequenced and has a short life cycle, so we don't have to wait like 50 years for like a tree to grow or something. It only takes like a month for Arabidopsis thaliana to get to its full capacity. We use a cabbage leaf curl because its growing point ceases development and the leaves wilt.
Alright, and then basically this is one of the, there's many groups of viruses in the Gemini virus category, and this is just one of the many that we decided to focus on, as Michael said.
Procedures. First, we had to purify the mRNA by grinding up the plant into something that we can use, and then we used the column method to nip.
Alright, so started with purifying the mRNA. So instead of going straight to the DNA, we had to use the messenger RNA. So we took that, and we had to make sure the messenger RNA was clean, it was pure. So first we ground it down so it was smooth, super small powder and stuff. Then we, in liquid, and added some extra stuff to make sure that there was nothing other foreign contaminants that would mess with our graphs and confuse our scales and stuff. So then with the column method, we ended up with the column method, that's continuing to purify as it's in the steps and moving it through filters. Then we ran it through gel electrophoresis to just make sure the integrity was still intact of the RNA. Then through the transcribing, the primers reversed the process, going towards DNA, and used reverse transcriptase.
Yeah, the reverse transcriptase is an enzyme that instead of like regular transcription, when DNA is changed to RNA, the RNA is changed back into DNA.
Alright. Quantitative polymerase chain reaction, or qPCR. This is basically, over here, we've taken the DNA, and the reason this leads back to why we picked this plant, how we have already, the full genome has been sequenced, so we know the specific DNA strand we want with the genes. So we take that DNA strand, we heat it up, so it all breaks, so like half the strand breaks down, We put in the machine puts the primers in, and it cools down, it heats up, and adds some other stuff, and then it goes back to the original DNA with extra DNA fragments that have been affected by the virus. So the way this is used is you put it into a machine, it goes through and does this 40 times. So 40 times it gets extra DNA fragments and genes, and then every time we can see what the virus did to the genes. So for 40 times we see every time what the virus has done. So it helps us examine and see what the virus is doing. This is one of the charts, so the qPCR results. The lines are the CT, basically it's the crossing threshold. That is where it's the most ideal time to look at the genes in the DNA to see, that's actually first when we first see it. Because what we do is we put in cybergreen, it's a chemical that starts to glow and allows us to really start seeing the changes in the DNA. So in these lines, the crossing threshold, so the second it hits the crossing threshold, then we are available to see it, and that's as soon as we want to start looking at the changes. And the tops change a lot, but we don't look at that, we look at the bottom.
This is a smaller, clearer chart of the one before. The UPC is our control, so it didn't go anywhere. The next three, they didn't show enough progress to be upregulated or downregulated because they are between 1 and -1. And the next two showed enough progress to be upregulated. And the ones on the side are the other genes that we tested.
And so that basically, with what I was saying earlier about the crops and trying to counterbalance, basically what we noticed is when the plants had the virus, these two specific genes they increased. The number of that amount of gene increased. So then that way we know in the future to make it immune, we lower the amount. So then it's back to equal levels and everything's fine. And that is what we did.